So, what if the history of human evolution didn't actually take, hundreds of thousands of years of this slow, grueling trial and error?
Right. What if that whole standard story is just, well, what if it's misreading the data entirely?
Exactly. I mean, the story where countless generations of early hominids had to, freeze and starve and essentially die out, just waiting for a random genetic mutation to fix a real
It's
problem.
a massive assumption.
It really is. So, welcome to another deep dive. Today, our mission for you is to explore this really fascinating, massive debate about the actual timeline of our planet and the biological engine that drives life on it.
It's a pretty profound debate. We're going to be analyzing two wildly different interpretations of the exact same physical evidence.
Yeah, the exact same rocks, the same fossils, the same human genome.
Right. And to do this, we've pulled together a really compelling stack of sources. We're looking heavily at a framework called the Unified Theory of Geologic History, or UTGH.
Authored by Daryl Waters.
Exactly. And to make sure we're getting the full picture, keeping it balanced, we are contrasting that directly with a comparative textbook called Earth. Two stories.
Which is great because it lays out the mainstream scientific consensus right next to the UTGH claims.
Right. And our objective today is to look at a highly specific question. If the standard framework uniformitarianism is mathematically correct, it requires an immense amount of depth.
An unbelievable amount of waiting around for random chance.
Yeah. So we want to see if this alternative mechanism proposed by UTGH, which relies on epigenetics and this concept of a front-loaded genome, could solve that rapid adaptation problem more cleanly.
And crucially, we're going to see how UTGH claims to solve these really stubborn scientific mysteries. I'm talking about things like giant flying dinosaurs or, trees fossilized straight up through solid rock.
Without relying on ad hoc fixes, it actually offers highly specific, testable predictions.
Which is the coolest part. But before we jump into the numbers, we just want to make our stance really clear to you listening. We are here as neutral guides.
Completely neutral.
Yeah, we are just exploring the arguments presented in these texts. We're going to unpack the math, the biology, exactly how the author lays them out.
We are not endorsing a specific viewpoint. It's a highly polarized debate, and our role is just to impartially report on the content. We want to let you weigh the evidence for yourself.
Exactly. So to even understand this alternative UTGH framework, we have to start by dissecting the standard model, right? We have to look at the math.
The foundational math, yeah.
Mm-hmm.
Mainstream science uses this framework called uniformitarianism.
Right, which from the textbook, my understanding is that it's essentially just the assumption of constant rates.
That's a good way to put it. It's the idea that the processes we see right now, like a river eroding a canyon or DNA mutating at a specific speed, those processes have always run at those exact same speeds.
Right. So if a river cuts a millimeter of rock a year today, it always has, meaning a 1,000-meter canyon took 1,000,000 years.
Precisely. They often call it the present is the key to the past. And when you apply that patient deep time concept to biology, you get the standard evolutionary model.
Where biological diversity formed super slowly over millions of years through random mutations.
Right. But the UTGH sources point out that when you actually run the numbers on human evolution, that math gets incredibly brutal.
Oh, it gets so brutal because the standard model says modern human genetic diversity, like all the traits and differences between populations today, requires at least 50,000 to 100,000 years of geographic separation.
Since humans migrated out of Africa, yeah. And the textbook actually breaks this down into generational arithmetic, which I think makes it much easier to visualize.
Oh, definitely. So let's use the 70,000-year mark as the baseline for when modern humans started migrating.
Okay, and if we want to know how many generations of independent evolution that is, we have to divide it by a generation time.
Right, and the text is pretty generous here. It uses 30 years for a generation, which, for ancient hunter-gatherers seems a bit high, but we'll go with it.
Right, so 70,000 divided by 30 gives you roughly 2,333 generations.
2,333 generations. I mean, just to put that in perspective for you listening, tracing your family back 10 generations puts you in the 1700s.
Right, so we are talking about a vast expanse of time. And over all those generations, the standard model relies on one specific microscopic mechanism to make all this diversity.
Random mutation, which is where the textbook really forced me to slow down and look at the constraints.
The mutation constraint is fascinating. The text states the standard germline mutation rate, measured right now in modern humans, is roughly 1.2 times 10 to the -8th mutations per base pair per generation.
Okay, translate that scientific notation for me. What does that mean for functional biology?
Well, think about the human genome. It has about 3.2 billion base pairs of DNA. So every time a cell divides to make a sperm or egg cell, it has to copy all 3.2 billion letters.
Like a massive molecular copy machine.
Exactly. And the enzyme that does it, DNA polymerase, is really accurate, but it makes typos.
Right, random copying errors.
So based on that exact mutation rate, every time a human reproduces, they pass on roughly 38 brand new novel mutations to their kid.
Just 38 typos in a manual of 3.2 billion letters. That is so small.
It's tiny. But under the uniformitarian assumption, if you isolate lineage for 2,333 generations, those typos slowly pile up.
They call them SNPs in the text, right? Single nucleotide polymorphisms.
Yes, SNPs. And the standard model claims that the observed genetic distance between the most isolated human populations today, which is about 178,000 of these SNPs, is completely made up of these novel mutations.
Just slowly stacking up over tens of thousands of years. But here's where the UTGH critique gets really sharp.
The biological friction point.
Yeah, because natural selection relies entirely on those random mutations, but the vast majority of them are totally neutral.
Right, they land in non-coding junk DNA and do literally nothing.
Or worse, they're actively harmful. They break a protein that was already working fine.
Precisely. Getting a beneficial mutation, a typo that actually creates a new useful trait to survive a new environment, is statistically incredibly rare.
And even if you win the lottery, even if a lucky hominid gets a great mutation, it doesn't just instantly save the species.
Because it's only in one individual.
Right. And this brings us to that concept of fixation from the text. Fixation is basically the finish line, right? It's when a new trait goes from being a weird quirk in one guy to being in 100% of the population.
It becomes the standard equipment for the species. But the math they attach to this process is staggering.
Yeah, walk us through that population genetics calculation from the text, because it blew my mind.
Okay, imagine a really beneficial mutation pops up in a population of 10,000 individuals.
Yeah.
And we give this mutation a selection coefficient of 0.01.
And 0.01 practically means what?
It means the individual with the mutation have a 1% greater chance of surviving and reproducing than everyone else.
Which doesn't sound like much, but in evolutionary biology, a 1% advantage is huge, right?
It is considered a very strong advantage. But even with that, the math dictates it takes approximately 4,600 generations for that single mutation to slowly outcompete all the other variants.
To reach fixation in a population of 10,000.
Yes, 4,600 generations.
So multiply that by our 30-year generation time.
Yeah.
You are looking at 138,000 years just to lock in one single beneficial trait.
It's a massive bottleneck of time.
Okay, let's just unpack this. It's like trying to rewrite a survival manual by randomly changing one letter every year. You have to wait hundreds of thousands of years for a sentence to accidentally make sense.
And then you have to convince everyone else in the tribe to copy your new sentence.
Exactly, but think about what that means for the hominids actually living during those 138,000 years.
That is the brutal reality of the mechanism. It's fundamentally a mechanism built on death.
Because if the environment gets colder or a new predator shows up, the population doesn't have 138,000 years to adapt.
No, they would simply go extinct. The UTGH framework argues this math requires an unfathomable amount of trial and error.
Generation after generation of early humans freezing, starving, failing to survive, all while just waiting for a random typo to magically fix their biology.
The author argues that this model just doesn't align with how complex organisms survive rapid environmental upheavals. And honestly, it creates a massive archaeological mystery too.
Oh, the Uruk problem. I found this to be one of the most compelling parts of the whole debate.
It really highlights the disconnect. If there were truly 2,333 generations of slow, gradual biological evolution, we should see an equally gradual archaeological record.
Right, an unbroken gradient of human progress stretching back 70,000 years. We should see campsites slowly becoming villages, primitive farming inching forward over millennia.
But the textbook confirms we absolutely do not see that.
Not at all. What we actually see is complex civilizations appearing suddenly, worldwide, in a super tight 1,000-year window, roughly 5,000 to 6,000 years ago.
Places like Uruk in Mesopotamia or Memphis in Egypt.
Yeah, Mohenjo-Daro. Suddenly you just have these massive urban centers with 50,000 people. You have written language, monumental architecture, legal codes.
Astronomy, mathematics, all exploding seemingly out of nowhere. It completely skips the thousands of generations of prerequisite trial and error.
So how does the mainstream textbook, Earth, Two stories. Explain that massive gap.
Well, it attempts to explain it by suggesting earlier populations just used biodegradable materials so they didn't leave traces, or that the gradual evidence is buried underwater from ice age sea level rise.
But UTGH pushes back hard on those, right? Calling them ad hoc fixes.
Exactly. UTGH classifies them as excuses generated purely to protect the assumption of deep time when the physical evidence is missing.
UTGH has a radically simpler answer. The reason we don't see 2,000 generations of buildup is because those 2,000 generations never existed.
Which means we need a completely different biological engine. If the slow grind of random mutation is too slow, what's the alternative?
Right, how did we get all this biological diversity? And this is where we get into the core thesis of the UTGH framework.
According to the sources, the immense diversity of the human race didn't take tens of thousands of years. UTGH claims it was produced in approximately 200 generations.
200 generations total, from a founding population to today.
And for populations that became geographically isolated right after their proposed global catastrophe, it compresses even further to roughly 166 generations.
That is just a whiplash-inducing compression of time. When I read that, I couldn't comprehend how they mathematically justify it.
It sounds impossible under the standard model.
Yeah, how do you get every skin tone, every bone structure, high-altitude adaptations, all in 166 generations?
To understand it, we have to look at their concept of the front-loaded genome. The text argues the founding populations that survived the catastrophe were genetically completely different from us.
Right, they didn't have the degraded, mutation-riddled DNA we have today.
No, they carried genomes with maximum informational fidelity.
And the text uses a really fascinating metaphor to explain maximum informational fidelity.
The library metaphor.
Yeah. The standard model views the early genome as basically a blank page. And over millions of years, random mutations slowly wrote new information onto it, letter by letter.
But UTGH flips that entirely. It views the original founding genome as a vast, fully stocked, comprehensive library.
Meaning all the genetic info for every possible human adaptation was already present in that founding group. Cold tolerance, varied pigmentation, it was all packed in there, just sitting latent.
It was unexpressed, but it was there.
So the obvious question is, if the book is already in the library, how does an organism actually pull it off the shelf and read it?
Right, because if you migrate to a freezing mountain range, you don't have 138,000 years to wait for a typo to give you thicker blood. You need it now.
And this is where epigenetics serves as the biological accelerator in the UTGH model.
Epigenetics is crucial here. It's the study of how environments and behaviors cause changes that affect how genes work.
But crucially, epigenetic changes do not alter your actual DNA sequence.
Exactly. They don't change the letters in the book. They just change how the cellular machinery reads the book.
Let's explain the chemistry of that a bit, because the text mentions processes like DNA methylation and histone modification.
Right. DNA methylation is basically the body attaching tiny chemical tags methyl groups directly onto the DNA.
And if a gene gets heavily methylated, the physical structure of the DNA tightens up, right? So the cell can't read it. It essentially turns the gene off.
Yes. And histone modification is similar. DNA is wrapped around proteins called histones, like thread on a spool. Modifying those histones can wind the DNA tighter, hiding the genes, or loosen it, exposing them.
Those are the epigenetic switches. So back to the UTGH timeline, the framework claims that the extreme environmental stress of the post-catastrophe world acted as a massive trigger.
We are talking sudden, massive spikes in UV radiation, drastic drops in atmospheric pressure, radical temperature shifts.
That extreme stress basically screamed at the genome. Rather than waiting for random errors, the stress triggered massive epigenetic cascades.
The switches flipped. The dormant variants in that front-loaded library were suddenly accessed and expressed almost immediately.
Just to ensure the survival of the organism. So the diversity we see today wasn't built over deep time. It was expressed in rapid time.
And what makes this so compelling is they point to real-world examples happening right now, like the African desert locust.
Oh, this example blew my mind, because the mechanics are unbelievable.
It's a profound demonstration of phenotypic plasticity.
Yeah, so under normal conditions, this bug is just a standard grasshopper. It's green, it blends in, it's physically small.
And it's behaviorally shy. It actively avoids other grasshoppers. It's completely solitary.
But if you take that exact same bug and subject it to environmental stress, specifically population crowding, a massive shift happens.
The trigger is entirely mechanical. When they get crowded, they physically bump into each other. That repeated tactile stimulation on their hind legs sends a signal to the brain.
triggering a massive serotonin cascade. And the physical transformation is basically science fiction.
It is extreme. Within hours, it starts turning a bright, highly visible yellow and black. Its body shortens.
Its brain literally expands and its muscles bulk up so it can fly long distances.
And behaviorally, it totally inverts. It goes from avoiding grasshoppers to desperately seeking them out, forming these massive, aggressive swarms.
And the vital takeaway here from the UTGH perspective is that absolutely zero genetic mutation happened during that shift.
Zero. The solitary green grasshopper and the swarming yellow locust share the exact same DNA sequence. It's the same genetic manual.
The environment just pulled a different, highly aggressive survival book off the shelf.
And the UTGH text applies this exact same engine of rapid adaptation to domestic animals too, to explain massive changes in short time frames.
Right, like domestic dogs. How do you get a tiny Chihuahua and a massive Great Dane from a single founding population of wolves?
Well, we know it didn't take millions of years of random mutation. Breeders did it in a few centuries.
The standard model acknowledges that, obviously, but UTGH focuses on how it's achieved mechanically. They didn't sit around waiting for novel protein mutations.
No, they differentiated rapidly through regulatory mutations and expressing existing variants. The text mentions the IGF-1 gene, which regulates body size.
Right. The specific variant of IGF-1 that makes a Chihuahua tiny wasn't a brand new mutation from breeding.
Sequencing shows that the small-size variant was already present in the ancient founding wolf population. It was just a low-frequency allele.
The genetic switches for small size were already loaded in the wolf genome library. Human breeders just applied intense environmental pressure.
Artificial selection, in this case. And the genome responded by expressing what was already latent.
So UTGH argues, if a wolf genome can produce every dog breed on Earth in a few centuries just by expressing existing potential, why couldn't the human genome do the same thing?
Especially since they argue the ancient human genome was far richer and undamaged. It's a logically consistent scaling of the mechanism.
But this is exactly where the math of standard population genetics clashes violently with UTGH. I have to push back hard on this next point.
The bottleneck.
Yes. UTGH claims the founding human population after this global catastrophe was just 8 individuals, 8 people.
Standard genetics dictates a bottleneck that severe is a guaranteed death sentence.
It's an immediate inbreeding disaster. The heterozygosity, the genetic variety would be so low, homozygosity would skyrocket.
You're talking about severe inbreeding depression.
Exactly. When a population drops to 8, they have to interbreed with close relatives, and when that happens, they pass down the same broken, recessive mutations.
Creating offspring with 2 copies of broken genes, leading to genetic diseases and rapid extinction.
We see it today with cheetahs. So how can the author justify an 8-person bottleneck without them dying out in 3 generations?
The UTGH defense rests entirely on differentiating modern, degraded populations from pre-event populations. Those mathematical extinction models are calibrated for our modern DNA.
Degraded meaning we've been accumulating 38 typos a generation for a long time. We have a massive reserve of broken genes.
Right, a heavy mutational load.
But UTGH proposes the pre-event world was a radically different physical environment, which altered the rate of genetic degradation.
They argue the Earth had a much denser atmosphere, right? Like 1.5 to 2.0 atmospheres of pressure. Yes, a hyperbaric environment with 30 to 35% oxygen and an incredibly thick canopy that blocked nearly all incoming UV radiation.
And UV radiation is a massive driver of DNA damage. It literally hits the DNA and fuses adjacent letters together, breaking the code.
So under those highly shielded conditions, the rate of DNA damage accumulation was effectively 0. The founding population of 8 didn't have a heavy mutational load. Their DNA was pristine.
Okay, so because the harmful typos simply hadn't been written into the code yet, the fitness penalty for inbreeding was negligible.
Right, 2 closely related people could mate, and they wouldn't pass on shared genetic diseases because those diseases literally did not exist yet.
Okay, that's the biological argument, but they also pair this with a mathematical argument about lifespan, right?
Yes, which alters the population genetics further. UTGH takes ancient genealogies as literal records, claiming early post-catastrophe generations lived for centuries.
Like up to 900 years. Okay, playing devil's advocate here. Biological immortality or centuries-long lifespans contradicts everything we know about cellular aging.
You're referring to telomere degradation.
Right. Every time a cell divides, the telomere, the caps on the chromosomes get shorter. When they run out, the cell enters senescence and the body dies. How does UTGH wave away the Hayflick limit?
The text links aging back to that environmental shift. They propose that in that near-zero mutational pre-flood world, telomere shortening was vastly reduced.
Or that telomerase, the enzyme that rebuilds telomeres, remained highly active.
Exactly. And when the atmospheric shield collapsed, the sudden influx of radiation and oxidative stress initiated the rapid telomere degradation we experience as aging today.
Okay, so if we just grant the premise of these long lifespans for a second, how does that actually solve the inbreeding math?
Because inbreeding coefficients, the mathematical danger of genetic overlap, compound by generation, not by year.
Ah, okay.
Think about a 1,000-year period. A modern population reproducing every 30 years will cycle through roughly 33 generations.
And with each of those 33 generations, the inbreeding bottleneck tightens and the danger skyrockets.
But if a population has individuals living for centuries, and they only birth a new generation once a century, in that same 1,000 years, they only experience 10 generations of compounding inbreeding.
So the compounding interest on that genetic debt is growing much, much slower.
Exactly. So combining a pristine genome lacking hidden diseases with an elongated generation time, UTGH argues an 8-person bottleneck isn't mathematically impossible.
It just looks impossible if you apply modern biological constraints to an ancient world that had different atmospheric physics.
That's their core argument.
All right, so if we assume UTGH's biological engine is structurally more efficient than waiting millions of years for random mutations, how does its geological mechanism compare?
This is a crucial transition. We have to look at how the standard uniformitarian model handles physical anomalies versus how UTGH unifies them.
And this brings up the idea of paradigm shifts. UTGH is highly critical of how mainstream science deals with evidence that contradicts deep time.
Right. The author argues that when the standard model hits a glaring anomaly, it generates an ad hoc fix.
Basically, a highly specific, localized exception just to protect the sacred millions of years timeline.
Let's look at the mysteries the text highlights. The first is polystrate fossils.
Breaking down the word poly, meaning many, and sucrate, meaning layers of sedimentary rock. These are fossils, mostly massive tree trunks, found standing vertically upright through multiple distinct rock layers.
Which presents a severe puzzle for the standard model. According to uniformitarianism, those layers were laid down incredibly slowly. 1 meter might be tens of thousands of years.
Which makes absolutely no mechanical sense when you're looking at a tree standing straight through them. If a tree dies today, it rots, it falls over, bugs eat it.
It doesn't stand perfectly upright, defying gravity for 1,000,000 years while dirt patiently piles up around it.
The top would decompose into soil in a century. So what is the standard model's ad hoc fix for this?
They invoke local rapid burial exceptions. Mainstream geologists argue that in that specific location, a highly localized flood happened, quickly burying that one tree.
But UTGH points out a fatal flaw there. There aren't just one or two of these trees. There are thousands of them on every single continent.
All embedded in rock layers that are otherwise confidently claimed to represent millions of years of slow deposition.
So you have to believe in millions of tiny, perfectly localized floods that magically buried thousands of upright trees globally while still pretending the surrounding rock is slow deposition.
UTGH argues it's an absurd pattern of coincidences.
So what's their solution?
A single, unified mechanism. They argue the layers were never separated by millions of years. They are the result of continental-scale hydraulic sorting during a single, massive rupture event.
The global catastrophe. Let's explain hydraulic sorting because the mechanics are fascinating. If you put dirt, sand, and gravel in a jar of water and shake it violently,
The sediments don't settle randomly. The laws of fluid dynamics sort them horizontally by density and weight. Gravel at the bottom, then sand, then fine silt.
distinct layers form in minutes, not millennia. UTGH argues massive, high-velocity floodwaters stripped immense volumes of sediment and redeposited them rapidly.
Sorting around the upright trees, burying them instantly before they could rot.
It also explains why the boundaries between those ancient rock layers are often so smooth and clean. There was no time for millions of years of weathering or animal burrows to mess them up.
It's a macro-scale mechanism replacing thousands of localized excuses. And they apply this same logic to biology, which brings us to sauropod biomechanics.
This is one of the most compelling arguments in the text. We're talking about the largest dinosaurs ever, like Argentinosaurus.
Biological leviathans weighing 70 to 100 metric tons, with necks extending up to 10 meters into the air.
And the physical problem is their cardiovascular system. To pump blood 10 meters straight up against gravity to keep a brain oxygenated requires immense blood pressure.
Biomechanical engineers calculate it requires about 700 millimeters of mercury at the heart just to deliver a trickle to the brain.
And to put 700 in perspective, look at the giraffe. The current biological maximum in a living land mammal tops out at 280.
And the giraffe needs a freakishly enlarged heart with massive walls, thick elastic vessels, and a specialized capillary net just to manage 280 without its vessels bursting.
Right, so 700 is an order of magnitude beyond biological limits. Normal blood vessels would literally explode. The heart walls would be so thick there'd be no room for blood inside the chamber.
It is biomechanically impossible under current atmospheric conditions. So what's the standard model's fix?
Because they refuse to change the atmosphere, they resort to behavioral adjustments. They propose these massive animals just didn't hold their necks upright.
They argue sauropods must have held their 10-meter necks perfectly horizontal to the ground, sweeping back and forth to eat low ferns.
Even though the skeletal structure, the vertebrae articulation, and massive ligament points clearly shows the neck was designed to sweep upward, they force the behavior to fit modern air.
What is UTGH's solution to this cardiovascular nightmare?
They propose a physical environmental mechanism. We touched on this. 1.5 to 2.0 atmospheres of pressure in the pre-catastrophe world.
And how does thick air solve a heart problem? Think of a modern fighter pilot pulling high Gs. The force pushes blood into their legs, threatening to starve their brain.
So they wear a G-suit. It tightly inflates around their legs, squeezing the body from the outside to physically force the blood back up.
Exactly. That external atmospheric pressure of 2.0 atmospheres acts on the sauropod like a global permanent G-suit.
The denser atmosphere compresses the immense venous network, providing massive hydrostatic assistance. It physically helps the heart push the blood back up the column.
And the math is stunning. At 2.0 atmospheres, with conservative estimates of hydrostatic assistance, the net cardiac requirement drops from an impossible 700 down to approximately 247.
Which is completely viable. It's well below the giraffe's 280. Suddenly, the whole cardiovascular system works perfectly. They don't have to sweep their necks on the ground.
They can browse the treetops normally, simply because the denser atmosphere provided the physical support they needed.
And this raises the third major anomaly, applying the same principle to aerodynamic flight, specifically the giant pterosaurs, like Quetzalcoatlus.
The largest known flying animal in Earth's history. It stood as tall as a giraffe with an 11-meter wingspan, weighing an estimated 250 kilograms.
The size of a small Cessna airplane. And the standard model has an insurmountable problem here. It's the aerodynamic lift equation, right?
Yes. Lift is dictated by fluid density, object velocity, and wing surface area. Getting 250 kilograms of biological tissue into sustained powered flight under current air density is fundamentally impossible.
Just look at the largest flying birds today, like the Andean condor. They max out around 15 kilos and need a running start off a cliff.
So how does the standard model fix the problem of a 250-kilo animal flying?
Once again, by creating an ad hoc behavioral reclassification. Because the math proves it can't fly today, some researchers just reclassified Quetzalcoatlus as a ground-stalking predator.
They claim it couldn't really fly, despite its highly specialized wing geometry and massive keeled sternum meant for anchoring immense flight muscles.
It was obviously built for vigorous flight, so the standard model looks at a dinosaur that can't fly today and says, well, it must not have flown.
But UTGH looks at the exact same bones and says, this animal is an instrument measuring a sky that doesn't exist anymore.
I love that quote. The biology is a recording of ancient physics. UTGH runs the lift equation backward, right?
Yes. They calculate that for a 250-kilo animal to achieve takeoff and flight, the air density must have been substantially higher. Specifically 1.3 to 2.0 times denser than our current atmosphere.
Which perfectly, elegantly aligns with the 1.5 to 2.0 atmospheres needed to keep the sauropod's heart from exploding.
That is what scientists call convergence. You have two totally independent biological constraints, internal fluid dynamics on the ground and external aerodynamics in the sky, independently deriving the exact same atmospheric requirement.
That kind of convergence is really powerful. But, you know, an interesting story isn't science unless it can be tested.
Falsifiability is the cornerstone of the scientific method. Hindsight is cheap. A robust theory has to predict exactly what researchers should find in the future.
And the author of UTGH lays his cards on the table. He outlines highly specific predictions based on this 5,000-year compressed timeline. Let's look at prediction 1, the Sequoia cores.
Right. UTGH proposes that the absolute oldest living giant sequoias in California are actual survivors of the pre-flood world.
That is a massive claim. So what's the physical prediction that proves it?
It comes down to dendrochronology, or tree ring dating. Tree rings are formed by the stress cycle of shifting seasons.
Rapid growth in the spring makes light wood, and slow growth in winter makes a dark, dense line. No seasons means no bands.
Exactly. Because UTGH posits the pre-flood world was enveloped in a thick atmospheric shield with no axial tilt, it claims there were completely uniform climate conditions, no winter, no summer.
So the prediction is that if you take deep core samples from the oldest sequoias, you will eventually reach a pre-seasonal zone of wood near the center that has absolutely zero annual growth rings.
A perfectly flat, uniform density profile. Now, mainstream scientists know about thick heartwood, where the center looks indistinct visually.
But UTGH goes further. Thick heartwood still has microscopic ring boundaries that you can see with X-ray densitometry. UTGH predicts the specific pre-seasonal wood will have zero oscillations whatsoever, a completely flat line on the scan.
And it gets even more specific geochemically.
Hmm.
The exact point where that ringless wood abruptly ends and the first true seasonal ring begins represents the exact moment of the catastrophe.
And because UTGH claims this rupture event expelled massive amounts of deep mantle material into the sky, it predicts an anomalous spike in the element iridium right at that specific ring boundary.
Along with highly altered strontium isotopes.
That is wild. They found iridium globally at the KT boundary, proving the meteor killed the dinosaurs. UTGH says you should find a similar spike, chemically locked in trees that are still alive.
And the best part is we don't have to cut down a protected sequoia to test it. Thousands of deep core samples already exist in archives. A geochemist could test it tomorrow.
If they find regular annual rings all the way to the center, or no iridium spike, the theory takes a massive hit. It's incredibly testable. Let's move to prediction 2. Private allele counts.
Back to human DNA. Let's define a private allele for the listener.
It's a specific genetic variant, a novel typo found exclusively in one isolated population, completely absent everywhere else.
It represents a new mutation that accumulated only after that group became reproductively isolated.
So using a family tree analogy, if a branch of a family moves to an island and stops communicating, any new unique traits in their kids belong only to that island family.
Precisely. If we sequence an isolated population, like Aboriginal Australians, we can count exactly how many unique typos they have. And the timelines generate vastly different math here.
The standard model says Aboriginal Australians have been isolated for roughly 50,000 to 65,000 years. At the standard mutation rate, that implies thousands of generations of isolation.
Mathematically dictating they should have accumulated tens of thousands of these unique private alleles.
But UTGH asserts they've only been isolated since the rapid dispersion after the catastrophe, roughly 166 generations ago.
So if we multiply 166 generations by the roughly 38 new mutations per generation.
The UTGH framework calculates they should possess only about 6,000 to 7,000 private alleles per lineage.
A staggering discrepancy. Tens of thousands versus a highly specific target of 6,000 to 7,000. And again, this isn't something we have to wait for future tech to solve.
Right. Massive open-source genomic databases like the 1000 Genomes Project have already sequenced these populations. We can just run the query.
If it counts are in the 30 or 40,000s, UTGH is falsified. If they cluster tightly around 6,000 to 7,000, it provides powerful confirmation of the compressed timeline.
Which brings us to the final test, prediction 3, subglacial Antarctic DNA. Which sounds like a sci-fi movie.
It does. It's based on UTGH's rapid migration model following the catastrophe. It suggests survivors scattered outward from the Near East very aggressively.
And because global sea levels and ice were radically different in the immediate post-flood ice age, UTGH suggests humans may have reached Antarctica before it was entombed in ice.
So if human remains are ever recovered from beneath the Antarctic ice sheet, what does UTGH predict about their DNA?
Well, standard models assume a slow creep over tens of thousands of years, so they'd predict any ancient humans found down there would relate most closely to foundational Near East or African populations.
But UTGH predicts their DNA will cluster tightly and specifically with modern Aboriginal Australians and other southern migration genetic isolates.
Because they weren't a slow creep, they were the absolute vanguard of the exact same rapid dispersion wave that populated Australia.
If the DNA clusters with ancient Near Eastern populations instead, the UTGH migration model is definitively wrong.
It's highly specific and easily disprovable, and I want you, the listener, to pause and consider why making these bold predictions is so vital. It comes back to convergence.
Right. If you calculate the fluid dynamics of sauropod blood pressure, the aerodynamics of a pterosaur, the specific tally of private alleles in Aboriginal DNA, and the lack of cellular rings in a sequoia tree.
And all of these vastly different physical parameters point to the exact same compressed time frame. It powerfully challenges the idea that all these different fields just happen to share the exact same wrong initial conditions.
Convergence among methods that share a common assumption is expected. But convergence among completely independent physical, chemical, and biological instruments demands a unified explanation.
It has been a wild ride through the mechanics of Earth's history today. We've explored a framework that looks at the Earth and the genome not as a slow, agonizing process of trial, error, and immense death.
But as a system of immense preloaded potential unleashed in a geologic instant.
We saw how epigenetics could serve as a rapid engine of adaptation, how a denser sky could let giants fly, and how this framework makes testable predictions.
It forces us to ask if the instruments we're using to measure the past are calibrated correctly.
And to close this out, I want to leave you with a final provocative thought, drawing on that genome metaphor.
The blank page versus the vast library.
Exactly. The standard model views your genome as a blank page that random mutations slowly wrote on over deep time. But what if your genome is actually an ancient, complete library?
The biological diversity we see today isn't the slow writing of the books. It's just the process of pulling different books off the shelf when the environment violently demands it.
So if that's true, if your genome is actually a vast, undamaged library inherited from an ancient world, what other unread books are still sitting latent in your genetic library, just waiting for the right environmental trigger to be expressed?
It's a profound biological question to ponder.
Thank you so much for joining us on this deep dive. Keep questioning the consensus, keep exploring the mechanics of the world, and never stop looking at the past with fresh eyes. We'll see you next time.